Hello students, we will be discussing antimicrobial resistance in two parts. So there will be two sessions of 30 minutes each around, and we will be discussing antimicrobial resistance, which is a talk of the healthcare community, the healthcare sector, and also of concern with the global governments and task force. And this is a very important topic for awareness and for necessary action for all the pharmacist community, not only in India but all over the world, in the interest of the human health. So let us see what is the timeline and discovery of major antibiotics and antibiotic resistance, how antibiotic resistance came into picture. And the if you see that this timeline of discovery of antibiotics, so in 1910, uh, salvarsan was the first antibiotic that was discovered in 1910, and in 1920 penicillin was discovered. In 1930 sulfonamides were discovered. So these are the three important discoveries of antibiotics: salvarsan, penicillin, and sulfonamides. And um, in 1940s, aminoglycoside antibiotics, tetracycline, bacitracin, and many other antibiotics were now coming up. And in 1950s, macrolide, polymyxin, nitrofuran, furan category of antibiotics were discovered. So 1940 and 1950 we witnessed the lot of development in the field of antibiotics, and they were discovered and they were available in the market as well. So in the mid 1950s, cephalosporins, quinolones, fusidic acid, gramins, ethambutol, cycloserine, all these sort of antibiotics came into the market. And then after, like within a gap of 10 years, in the mid 1960s, phosphenoids were introduced, and in mid 1970s, around 1975, the bacterian carbapenems and mupirocin, these were the new antibiotics that were floated into the market. In 1980s, fluoroquinolones, which are the most important antibiotics we consider very effective. 1980s, around 1980s, fluoroquinolones came into the market. And in 1990s, lipopeptides and oxazole phenones, they are all discovered. So we can see that major discoveries happened in the area of 1940 to 1960. Lot of antibiotics were discovered during that time. And but as we now move forward, after 2000, we hardly witnessed any new antibiotic coming into the market. So the term antibiotic resistance, it came up because in 1910 salvarsan was discovered, and around 1930 salvarsan resistance was also discovered. Similarly, in the late 1920s, sulfonamides were discovered, but in the early 1930s, sulfonamide resistance was also seen, witnessed. In 1920s, the penicillins were discovered, but within a short span of time of 20 years only, penicillin resistance was also witnessed. Similarly, a similar graph was observed for all other antibiotics like tetracycline, MRSA, gentamicin, vancomycin, and carbapenems, and etc. So we can say that uh, as soon as antibiotics are available in the market, within a span of 10 years, average 5 to 10 years, the resistance is also observed for the same antibiotic. So this is just to give you a brief idea. But now there is a common term that is trending all over the world, which is known as superbugs. So what are superbugs? So basically superbug refer to the germs, or germs yani pathogenic microorganisms, that have shown resistance to antimicrobial agents used to treat them, and include multi-drug or pan-drug resistant bacteria and fungi. So superbugs are those pathogenic microorganisms which show resistance to the antimicrobial agents or antibiotics. So the common, you can say the common ones are the terminology that is used for the group of six highly famous superbugs is escape, which stand for enterobacteriales, staphylococcus aureus, klebsiella pneumoniae, acinetobacter baumannii, Pseudomonas aeruginosa, and enterobacter. So these are the group of six microorganisms which are they are considered to be the superbugs. Then there is a carbapenem resistant enterobacteriales. Then there is a CRKP, that is carbapenem resistant Klebsiella pneumoniae. Because carbapenem, we know that they are mostly given for the upper respiratory tract infections and lung infections, pulmonary infections. So pneumonia is also the Klebsiella pneumonia is the bacteria that is responsible for pneumonia. Carbapenem is a widely used drugs and acceptable drug for that indication. But now we see that carbapenem resistant enterobacterials as well as carbapenem resistant Klebsiella pneumoniae is there, known as a superbug. Next is the methicillin resistant staphylococcus aureus, which shortly known as MRSA, that is also considered as a superbug. The next superbug is ESBL producing enterobacterials. The next one is vancomycin resistance enterococcus, multi-drug resistant Pseudomonas aeruginosa, and multi-drug resistant Acinetobacter. So these are all the examples of superbugs, which we call them to to indicate that these have evolved genetically over the years, and they have develop their defenses around the highly potent antibiotics. So when the antibiotic is popular, the microorganisms are intelligent enough to develop methods to fail the antibiotic. The next is the mechanism of drug resistance. How drug resistance occurs. So antibiotic and bacteria coexist in the same ecological niche, because antibiotics, the bacteria are, first of all, the bacteria thrive, and then we are introducing antibiotics to kill that bacteria or kill that microorganism. So basically it should also be able to exist in the same ecological niche. So the both are somewhere sharing some common features, and bacteria develop defense against the harmful effects of the antibiotics. So it is like a warfare, sort of chemical warfare that the bacteria is engaged into. Now if we consider the antibiotics to, what are the mechanisms that antibiotic use to harm the bacteria? There are four mechanisms. Either they destroy the cell wall of the bacteria, or they destroy the cell membrane, or they destroy the protein synthesis, they hamper the protein synthesis process, or they hamper the nucleic acid synthesis process. So these are the four ways in which antibiotics act to kill the or harm the bacteria. So similarly, there are four mechanisms of resistance development that are used by the bacteria against the antibiotic. The four mechanisms are limiting the drug uptake. So they will, their cell wall will not realize the entry of the antibiotic cell into the cell. So limiting the drug uptake. Second is altering the drug target, the protein or the drug target where the antibiotic is supposed to combine with. They alter the drug target. Next is inactivating a drug, and last one is, the fourth is increasing the drug active drug reflux. So these are the four ways by which the superbugs develop the resistance. Now let us see what is the fact sheet available with us. What is the data, what is the information, the basic information that is available. So WHO, World Health Organization, has declared that antimicrobial resistance is one of the top 10 global public health threats facing the humanity. So it is very serious concern around the globe by all the countries of the world. Now, the term antimicrobial, it includes antibiotics, antivirals, antifungals, antiparasitics, and all those medicines which are used to prevent and treat infections in humans animals and plants. So antimicrobial is a broad term encompassing antibiotics, antivirals, antifungals, and antiparasitics. Antimicrobial resistance occurs when bacteria, virus, fungi, and parasites change over time, maybe genetically or behaviorally or enzymatically, and they no longer respond to medicines, making the infections harder to treat and increasing the risk of disease spread, severe illness, and even death. So this is the antimicrobial resistance, the definition of antimicrobial resistance. So when the drug is no longer effective, or no longer, the bacteria is no longer responding to the drug and or the medicine, and that is why it becomes harder to treat the infections. A new Sustainable Development Goal indicator for AMR was approved in 2019 for bloodstream infections, means systemic infections due to specific drug resistance pathogens. So this will facilitate global monitoring of the spread of antimicrobial resistance as well as the effectiveness of intervention. So a new indicator, a mechanism, a measuring was developed uh, as a sustainable development goal indicator for AMR, that is 3.D.2. So healthcare, in the category of healthcare, in the SDG of healthcare, another indicator was introduced as an indicator for AMR, and it was approved in 2019 to address the hard pressed issue of antimicrobial resistance. Now, what are the drivers of antimicrobial resistance? What makes antimicrobial resistance difficult, and what gives birth to antimicrobial resistance, with what feeds the antimicrobial resistance issue is, the biggest thing is the misuse and overuse of antimicrobials. So we know that physicians and even patients, we all indulge into self medication, and physicians will also prescribe. There are reported cases of over prescribing of antimicrobial drugs or antibiotics, and even for antiviral, like viral fever, antibiotics are prescribed sometimes immediately. For a symptomatic fever, antibiotics are course is started. So there is a misuse and overuse of antimicrobials, and even when the symptoms of bacterial infection or fungal infection is not precipitated well, but still, as a precautionary measure, antibiotics are prescribed. So maybe it is a fungal infection, maybe it is a secondary infection, or maybe it's an indication of some other disorder or problem present in the body, but without giving the time, the duration of time, uh, there is a prescription of antibiotics. Lack of access to clean water, sanitation, and hygiene for both humans and animals. So we know that the clean drinking water and clean water is already a problem all over the globe, and there is a lack of availability and accessibility to clean water, sanitation, and hygiene facilities. Poor infection and disease prevention and control in healthcare facilities and farms. Poor access to quality, affordable medicines, vaccines, and diagnostics. Lack of awareness and knowledge, and lack of enforcement of legislation. So these are the various factors that give boost to antimicrobial resistance. They drive the antimicrobial resistance forward. Now, if we see the drug resistance in bacteria, so we know that now drug resistance is present in the bacteria, in the fungus, in the parasites. So let us see one by one the drug resistance in bacteria. For example, ciprofloxacin is a drug. Now the bacteria E. coli and Klebsiella pneumoniae is resistance, and the person, these are all evidenced, reported figures that I have collected, and percentage resistance reported is varying from 8.4% to 92.9%. So this against E. coli. Now E. coli is such a bacteria that it is present inside our body also. It's a naturally present, available in the microbiome of individuals, and but when it grows up beyond certain number, it causes infections and it causes certain problems. So to treat that UTI, and ciprofloxacin is mostly used, and it was a drug of choice, but unfortunately a high good percentage of resistance is seen with E. coli infections treated with ciprofloxacin. Similarly, for Klebsiella pneumoniae, that is ciprofloxacin was also used in the URTI as a drug choice, choice of drug. But for K pneumonia also, the percentage resistance that is reported is from 4.1% to 79.4%. Similarly, for carbapenem, which is a drug of choice for upper respiratory tract infections, especially Klebsiella pneumonia, the percentage resistance exactly I could not find, but there is a wide, reporting of resistance of Klebsiella pneumonia against carbapenem. Now, the third generation cephalosporins, MRSA, that is methicillin resistant S. aureus, Staphylococcus aureus, the bacteria is found, and this bacteria species, the strain is resistant. The percentage resistance is around 12.1% for the third generation cephalosporin. And for against this drug, the E. coli is also, the strain is resistance up to 36% of E. coli resistant strains are found against the third generation cephalosporins. Similarly, for the species N. gonorrhoeae is also found now resistance towards sulfonamides, penicillins, tetracyclines, macrolides, fluoroquinolones, etc. And even fluoroquinolone antibiotics, E. coli has evolved itself, and it has now acquired the resistant version of itself against fluoroquinolone antibiotics as well. So the major antibiotics, which are the antibiotic of choice for a large wide variety of people around the countries, now the bacteria have developed themselves and they are now resistant to these common drugs. Now, drug resistance in Mycobacterium tuberculosis. So, WHO estimates in 2018 there were about 500,000 new cases of rifampicin resistance TB. So, rifampicin and INH were the two, majorly used tubercular drugs. But now we have this strain of M tuberculosis, which is resistant to rifampicin also. So that is known as RRTB, and it is globally present, and that is why now rifampicin is not that highly used, and that used with confidence of using the rifampicin is low now. So multi-drug resistance tuberculosis requires treatment courses that are longer, they are less effective and far more expensive as compared to the routine ones, because it takes a lot of time to treat such infections, and lot of combinations of antibiotics need to be provided to the patients. So less than 60% of those treated for multi-drug resistant RRTB are successfully cured. Only so there is a 50% chance that you will be cured successfully without any relapse. In 2018, an estimated 3.4% of new TB cases and 18% of previously treated cases had multi-drug resistance TB. And the emergence of resistance to new last resort TB drug to treat drug resistant poses also a major threat. And now we don't have many tuberculosis drugs in pipeline, and lot of research is going on globally to address this issue. Now, if we see the drug resistance present in the viruses or reported in the viral diseases, so level of pre-treatment in case of HIV drug resistance to non-nucleoside reverse transcriptase inhibitors among adults initiating first-line therapy, therapy exceeded 10% of the majority of monitored countries in Africa, Asia, and Latin America. So, in sub-Saharan Africa, over 50% of the infants were newly diagnosed with HIV carry a virus that is resistant to NRTI, that is the non-nucleoside reverse transcriptase inhibitors like stavudine, zidovudine, and these type of drugs. Latest WHO ARV antimicrobial resistance guidelines for virals now recommend the adoption of new drug, that is dolutegravir, as a preferred first line treatment for adults and children. So even if we see, then the drug resistance is um, prevalent in malarial parasites also. So artemisinin based combination therapies, that is known as ACTs, are the recommended first line treatment for uncomplicated P. falciparum malaria and are used by most malaria endemic countries. So partial resistance to this artemisinin and resistance to a number of ACT partner drugs have confirmed in countries like Cambodia, Laos, People's Democratic Republic, Myanmar, Thailand, Vietnam, etc. From a 2 decade long studies. So partial resistance is also found against artemisinin for malaria parasite. So P. falciparum resistance by to sulfadoxine pyrimethamine led to artesunate sulfadoxine pyrimethane failures in some countries. So is even these combinations are also failing. So P. falciparum is also developing itself, and to combat to these antibiotics. In Africa, evidence has recently published showing emergence of mutations linked to partial artemisinin resistance in Rwanda. So we can see that there are reports, indicating that the even for the malarial parasite also the drugs are not effective, and resistance has been developed. Now, if we see drug resistance in fungi, so Candida aureus, drug resistant Candida aureus. So, not much research has been carried out in this sector, but drug resistance Candida aureus is one of the most common invasive fungal infections, is already widespread with increasing resistance reported to fluconazole, amphotericin B, voriconazole, as well as emerging caspofungin resistance. So Candida aureus was studied, and it was found that these common antifungal agents were now not no longer effective. Even caspofungin drug is also reportedly, the fungi has developed resistance towards this drug as well. The prevalence of drug resistance antifungal infections is increasing and exasperating the already difficult treatment situation. So already it is difficult to treat fungal infections because they first of all the treatment line is very time consuming, and fungus keep on changing. It is generally topical and difficult to identify, difficult to be targeted, by the antibiotics, antifungal agents, and it was already difficult treatment, but still it is now the fungus is also developed resistance towards these antifungal agents. So we know that what are the drivers to AMR we have discussed primarily. Again, just to sum up, so misuse and overuse of antibiotics, increase in gross domestic product, because the more and more use of antibiotics require more and more manufacturing of antibiotics, and that is why the gross domestic product GDP increases. So inappropriate prescribing patterns, so because the people are working, the manufacturing sector is increasing. So since people are now exposed to different type of conditions, so they acquire infections, and that is why it also causes the AMR. Inappropriate prescribing patterns, as we already discussed. Paucity in futuristic antibiotics, because we can see that not many antibiotics have been discovered after 1970s. So from 1940 to 1970, lot of antibiotics came into the market, but after that only a couple, one or two are only coming. There has been a stoppage, or a gap in the development of antibiotics, the drug discovery and development. Then even in the agricultural use of antibiotics, we see that as we are now dependent for dairy, for meat, fed poultry on the animals and farm animals. So to ensure their health and to ensure the availability of food in large amounts, even antibiotics are used there, and these antibiotics precipitate in the food products, even honey also. And this type of antibiotics we are consuming through food, through meat, through eggs, through honey, and they are going in our body. So we our body is exposed, the physiology is exposed to these antibiotics prima facie. So when the bacteria now infest us, these antibiotics are no longer effective. Then easy travel routes. So because of the shortage of travel time and the ease of travel, air travel, ship travel, even road travel, so people become the carriers for the antimicrobial resistant bacteria. So a person may come often from Africa to India, or maybe US to India, and so on. And these resistant strain of bacteria may persist up to 12 months carried in the body after a person has traveled to a highly endemic AMR regions, amplifying the risk of transmission among susceptible populations. And of course, there is a knowledge gap which contributes towards the antimicrobial resistance. So after this, uh, we will take a break and we will continue the same lecture in the second session of my podcast. So thank you everyone.